Some of the information in this Patterns of Evolution chapter we've covered in other chapters, or at least the ideas, and also a lot of times at the beginning of the semester we watch a film or some set of films, like What Darwin Never Knew perhaps, and that has some of these ideas in it as well. The first thing to recognize is that when we talk about evolution, it can happen rapidly within a couple generations. You can see allele shifts for sure, the microevolution. But a lot of times we're talking on what we call deep time. So deep time just means really, really long time ago. And it's hard for us to actually imagine because our time scale is so different than deep time. So what I wanted to point out here is that when we talk about billions of years, which is when we talk about the age of the Earth or the origins of organisms or cells and then prokaryotes, etc., we're talking on billions. So a billion is 1,000 million. And to put that into a context that might make more sense to you, a billion seconds is 31.7 years. And if you were trying to spend $1 billion in 1 year, you'd have to spend $2.7+ million a day. So a billion is a very large number. And when we talk about something like 4.6 billion years, the age of the Earth, we're looking at a really deep time scale. So that's something you have to keep in mind when these speciation events we're looking at, and you think, well, that seems like a pretty big change. First of all, we know that genetic programs that are in development can change with relatively little genetic change with those mutations and switches, but also that we're looking on deep time scale. A lot of times to figure out the history of life, we look at fossils. So there's all different sorts of fossils. Some of them are things like this, which are deposits left by particular types of organisms. Sometimes we have impressions. Sometimes we have the fossilized bones. You can even have non-animal fossils for sure. So sometimes we only think of animals, but plants can leave impressions that could be fossilized. This is an early eukaryote fossil. And so you're not going to need to know any of these dates for these fossils, but we're going to see how those come to be very briefly. To look at fossils, they look at what they call the strata or layers. And the idea is very simple here, that if you've cut open the Earth and you're looking at the Earth and you have layers of the Earth, fossils that you find deeper in the Earth are probably older than the ones you find shallower in the Earth. The only times we see that when we see that being different, it's because the geology of that structure has changed, like there's been a folding or inversion sort of event. But usually we can look at the geology and tell that that's happened. So we use positioning for relative dating because a lot of times we look for certain minerals in particular layers that we look for different elements in different layers that are radioactive. And so then we can figure out the age of that layer, and then that will tell us the age of the fossils. And so the radioactivity matters because it helps us do what we call radiometric dating. And you've probably heard of the carbon-14 dating. So carbon-14 is an isotope. Remember, isotope just means different number of neutrons, and some isotopes are radioactive because they are unstable, and radioactive means that they basically give off particles at a particular rate over time. And so the carbon-14 changes into a nitrogen type atom, and we can measure the amount of carbon that's left from when it started. And that's what this thing called a half-life is. So you notice that each time frame, the amount of that radioactive carbon, the fraction of it, reduces by half of what it was in the previous time frame. And so when we know the amount of time that it takes for a certain isotope to degrade, which is a half-life, then we can calculate the age. So carbon-14 is used for relatively recent fossils. There are other isotopes that are used for much older fossils, and in that case, you just have a half-life. Instead of 5,700 years, you'd have one that had maybe 1,000,000 years or something like that. We don't find fossils of every organism, and this is not unexpected. Fossilization is a rare process. It requires a lot of conditions to be in place. So the organism has to not get decomposed before it can fossilize, or at least the imprint can't go away. There has to be, usually there's some sort of water or running water. It happens only in certain rock types. And so when people are concerned that we don't have fossils for every, missing link, which I prefer to call a transitional species, that's not unexpected at all. We find very few fossils, and not only are the fossils being made rare, then we have to discover them. And so that all those combined make it pretty amazing, really, that we have as many fossils as we do. We have also an overrepresentation of certain types of organisms, the ones with the hard parts. So we have to keep that in mind when we're trying to figure out the diversity of organisms at any given time in any given area, that we're probably getting a bias towards those hard-parted organisms, like ones with shells or armor or bones, things along those lines. Here is a basic time of the Earth chart. So it starts here, and then this is modern day. And you can see that they have lots of cool information here. This is from HHMI Biointeractive, and you can actually download this Earth Viewer app on a tablet or a phone, as well as they have one that's in HTML, so you can go through the internet for that. And you can look at the Earth at different time frames and what's happening. It's pretty neat. We'll look more at the history of the Earth in regards to organism arisal as we go through the semester. But what I want to point out here is that the Earth's age down here, and these are the kind of numbers you need to know for this point, for this section. So the Earth is 4.6 billion years old approximately. So that's where we start at this end here. We see the oldest signs of life somewhere between 3.5 to 3.8 billion years ago. I'm going to skip a bunch of events in between because we'll talk about those later. But I want to point out that this is present day. Also that the initial organisms here, those are going to be prokaryotic organisms. We don't get eukaryotes arising until 1.8 billion years ago. So that is when the first eukaryotic fossils are found. Probably would arise a little bit before that. And then we don't really get animals, until much later down here, the Cambrian explosion, there it is. That's the diversification of animals and so forth. So you can see that things we think of as really old, the dinosaur extinction is right at the very end here too. Let me show you a close-up. So look at that. We think of dinosaur extinction happening like really long time ago. There it is on this scale. So this is a very recent event. And then here's the genus Homo, which is our genus. And that's not even Homo sapiens, which are even further down the line because there are other Homo hominins in the genus Homo that arose before Homo sapiens were there. So if we were to take a clock and split the history of life into, an hour, the existence of humans in that time frame would be less than 1 second. And you can see that here on this scale. And that's what we mean by deep time. So there's our 4.6 billion years that I mentioned. The oldest fossil organisms, like I said, somewhere between 3.5 and 3.9 or 3.8 billion years ago. And they were prokaryotic fossils, single-celled organisms, bacteria-like. And we'll talk more about them in the bacteria and prokaryote chapter. But they left these types of patterns in the rocks. Those are deposits from, we call those things stromatolites, and so that's how we know that they were there with additional evidence. So you'd want to know these dates and that date. I should also point out that when we look at fossil organisms, we can sometimes get misled about how much diversity there is in a lineage. So lineage A, and then there's lineage B. Okay, so common ancestor splits. And if you look at modern day, you'd think there's only one species in lineage A, and there are 1, 2, 3, 4, 5, 6, 7, 8 species in lineage B. So you might think that, wow, lineage A hasn't changed over time at all, and lineage B, it's branched and branched and branched. But that would be a conclusion you can't make, because extinction events happen. So that's what these little cross-looking things mean. So that's an extinction of a lineage there. And so, sure, this one branched lineage A. It's just that only one of the organism or one of the groups of that lineage is still living at present day. And so don't be misled by the fact that there's few of the organism type left today. It doesn't necessarily mean it's not a diverse clade in the past. It just could be extinction events, and there's only a few left. One of the ways we have speciation occur is that allopatric speciation, which is where one population gets split into two, and then each of those populations diverge. And some of that's island formation and things like that, but also the continents move. So here is a cross-section of the Earth. The crust is what we live on the outside. Then you have the mantle, which is a viscous sort of structure here. So basically the crust floats on the mantle, and the mantle moves around, and the crust moves around, and it's in these plates. So they're independent of one another, and you can see that the plates all have junctions. And because the plates are moving, sometimes they're moving apart on the mantle, sometimes they're moving along the other one, sometimes they're moving together. Like this Indian plate's moving into the Eurasian plate. And so those how we get different sorts of geologic features, for example, the Himalayan mountains. And you can also have all the volcanic and earthquake activity. So we know California, Oregon area, Washington State, that's all earthquake prone, and there's a lot of volcanoes like up in Oregon and Washington State, and that's because there's a plate there. You call them faults, right? The junction between two plates. Anyway, because these continents and the plates have moved around over time, sometimes they crash into each other and then they become one body. So actually you can see South America and Africa, you can see how this fit into there previously, and then they moved apart from each other. And so when they move apart, it creates an allopatric speciation scenario where some of that organism that used to be on that land mass ends up in the South American side, and some ends up in the African side, in the African plate, and those diverge differently. So we see common ancestry between certain organisms in both these areas, but then this speciation would occur because of the different selective pressures and founder effect and all that kind of stuff. One of the consequences of this continental drift with the plates moving around, I mentioned that sometimes they crash together and they form these megacontinents. And so we've had like 3 times in history where we've had all the land masses just in one big mass. You've probably heard of Pangea, but that's when it's here. There's also 2 other times that that's happened. And if you have the landmass all come together, then you lose coastline. So this changes the whole environment that the organisms on that landmass are exposed to, which would lead to speciation, macroevolution, that sort of thing. So what happens when those supercontinents form? You get a lot less coastline. It becomes drier inland because it's further away from water, and water, remember, is like the temperature moderator. So it's going to be drier and cooler inland than it was when they're all separate. You're also going to have a lot less shallow water. Mass extinctions are events when you have a very large portion of the organisms on Earth being wiped out. So the diversity plummets during those events. And most of the time this comes from some massive change or relatively for deep time, a short period in which a very large change in the environment happens. So for example, this last mass extinction here, that's the one that's the dinosaurs. And that mass extinction was caused by an asteroid hitting the Earth, actually by the Yucatan Peninsula. And basically what ended up happening was the sun was blocked out from all the debris, and the plants died, and then the herbivores died, and the carnivores, and there's a bunch of other stuff going on with the gases in the atmosphere and that kind of thing, and it led to this mass extinction here. And you can see that the diversity, which is in the, they're showing with the blue the number of families of organisms, dropped here and then has risen again. But you're like, okay, well, it's come up again from the dinosaurs. Sure, but it's still, we look at here, a million years ago, I mean, we're still looking at, this is 65.5 million years ago, and now we're at 0. So 65.5 million years is what we're looking for this increase in diversity. So, you know, it's a long process typically to recover from these mass extinction events, and only some of the organisms make it through. So these are changing the landscape of what living organisms there are on Earth. You can see there are these other mass extinctions as well. And here's the Permian extinction. That was a really, really big extinction event. And we know from looking at fossil or earth cores and things like that, that we had large amounts of CO2 from volcanoes. And so that, of course, we know increases the temperature because it acts as a blanket, and so then we have warming events, and so that was probably one of the big things that led to this Permian extinction, which was massive, 96% of marine animals. And then the Cretaceous extinction, that's the dinosaurs involved in that one, 50% of all marine species. There's the Chicxulub crater, so it's right over by us. Actually, if you go to Texas at Brazos River, can't remember, but there's evidence, like real, very clear rocks that don't belong there that came from the asteroid impact. And so it's kind of really neat that we're so close to all this evidence that's really big deal that told us how that dinosaur extinction happened. And as you probably know, we're concerned about the loss of diversity today. And so the question is there a mass extinction that is on the brink of, or are we in a mass extinction kind of event? And remember, when we see extinctions happening at a large or a high rate, even if it's just like 1% more than we expect, it's a really big deal because our time frame is so tiny. We don't see deep time. And so when we're seeing extinction rates rise, that means they're really rising a lot in the context of a deep time event. We also know that the overall warmer temperatures are correlating with the mass extinctions, and we know that our climate is warming. And remember, climate's different than weather. Weather is, you know, how much precipitation there is in a given day, that sort of thing. It's like a small scale. Climate is like overall, what does the area experience? And so it's a much longer kind of scale. So even though a place might get cold and hot within the progression of the year, we're looking at overall trends of warming, and that's very clear that that is happening, and it's very clear that the majority of that is because of the increase in greenhouse gases that are released to the atmosphere from a variety of sources, including fossil fuel, as well as livestock. They put off a lot of methane, and so, you know, driving our cars, a large part with the electricity, where we get our electricity from. Of course, you have to have energy in order to make energy or convert it to other energy, and so we get our energy from those carbon compounds. And then once that's broken off to get the electrons, you have carbons and oxygens left over, and that's what makes the CO2. So that's why we're so worried about it getting warmer. Not only it's going to be like potentially uncomfortable, if you will, but also that we see the trends before, we've looked at the data, we're looking at the data, and it's just not, it's looking like we are hurtling into an event that's similar to what we've seen in the past. All right, I mentioned that the loss of diversity and that it takes quite some time. So 5 to 100 million years to recover that diversity. And yes, it comes back, but most of the organism types that were there don't. And after the dinosaur extinction, this is when mammals really took over. So there were only very limited mammals when the dinosaurs were around, sort of like shrew-like type mammals, lived underground. But once the dinosaurs were gone, and many other types of organisms, those mammals now had niches to move into. So they had no less competition for space, for food, for places to raise their young, all those kinds of things. And so that's this thickness here. This is showing the number of species. So you can see that the number of species really exploded. And so this is what we call an adaptive radiation event, which is like with the birds on islands sometimes, or organisms on islands, when you see lots of diversification very quickly. That's what we're seeing with the mammals. So how would you get something like adaptive radiation or any kind of macroevolution happening? Well, of course, we know it's genetic changes. What we see very frequently is the changes in these switches or these homeotic genes, and these guys are what we call body plan genes. So basically those are going to code for transcription factors. And those transcription factors set up the body plan, which is just where the legs are going to go, where the head is, where the tail is, what's the back, what's the belly, where the antenna, all that kind of stuff. And that is, so then the transcription factors, remember, they bind and turn on multiple genes, and then those could be transcription factors too and turn on other genes, and eventually you get to the stuff genes, and those are actually going to either do chemical reactions, they're enzymes, or they're going to build things, or they're actually going to alter how the organism looks. So the stickleback story that you saw some of in What Darwin Never Knew, if we watch that, the expression of PITX1 is what they're staining for here in the purple. They've stained for that gene expression there. And you can see it's expressed in what will become the mouth, as well as these pelvic fin regions. So that's going to, or pelvic spikes. So that's going to make those spikes on the stickleback. And you can see that because of mutations in switches, or in the body plan genes, the switches, the switches that turn off and on body plan genes. So one of the switches was mutated, which means that PITX1 is no longer expressed in the ventral side, the belly side of the fish. But it's still expressed in the mouth, and actually it's still expressed in the brain and the pituitary gland. And so you can see that you get no, you'll get no stickles on this stickleback fish, but you still get the proper development of the brain and the mouth area because the expression still happens there. So the fact that we have multiple switches that control the same gene in different body regions means that mutations in one switch can change just one part of the body without changing other parts of the body. And this doesn't, this is just, how could you completely get rid of pelvic fins? Well, PITX1 is a transcription factor. It turns on other genes, and then those genes are either stuffed genes or they turn on other genes, and then that is what builds the pelvic spikes. And when PITX1 is, when the switch is mutated, it doesn't get turned on in the ventral side, which means that there is no transcription factor, so those genes don't get turned on that are downstream, and so you don't get these pelvic spikes. Heterochrony is this rate or timing of expression. We talked about that too if we watched What Darwin Never Knew. In the finch beak, and this can be more drastic as well, but in the finch beak, and the finches in their beak, they talked about that the gene was turned on in the 4th day of development versus the 5th day of development, that kind of thing. So that's the timing of expression, and that changed the beak depth. So whether they had a fatter beak or a skinnier beak. You can also change the rate of expression, so that basically means turn off and on the gene more or less, and that can change, it'll change the genetic program, because you change what transcription factors are on when and where, and that changes everything downstream, which eventually changes stuff genes or their expression. And so that's what leads to these body type differences. One type of homeotic gene are those Hox genes. So homeotic genes, one type in animals are the Hox genes, and so different Hox genes are expressed in different regions of the body. And which Hox genes are expressed where, when, and how much determines what structures are built there. And it's even which combinations of Hox genes. So sometimes there's 2 being expressed in one area or 3 being expressed, but if only one's expressed, it's different, which makes sense because they code for transcription factors, which then turn off and on many different stuff genes downstream. And that builds different body parts. I mentioned this in another video, is that evolution is a tinkering event, right, where it is a modification of previously existing structures, previously existing genetic programs and genes. And so we're looking at modifications. And we say slight modifications, so sometimes those phenotypically can be kind of drastic, like having an eye and having no eye. So there's a gene called PAX6, and if you knock out PAX6, you get no eye anymore. And that's one genetic change that creates no eye. So you might think, wow, losing eyes, that's a big deal, but it's not a big deal at all. And actually the PAX6 gene, if you put a mouse gene in a fly, it makes a fly eye. If you put a fly gene of the PAX6 in the mouse, it makes a mouse eye, which is actually really incredible because mouse eyes and fly eyes are not the same structurally. But the PAX6 is homologous enough, and then with all the other programs in the fly versus the mouse, it actually makes the correct basic structure of the eye in that organism. So we see that's clearly common ancestry there of that gene. We must go back in the animal lineage quite far. We're looking at multiple steps to get complex structures, and it's not always that complex structures just arise for, quote, a function, right? Like the flagellum, the bacterial flagellum. So it has a bunch of different parts. There's the cell, there's the flagellum, and then it has it's like a collar and some other proteins, a bunch of stuff there, and you're like, well, that's so complex, how does that arise? Well, actually, if we look at the proteins in the membrane, some of them are analogous, or homologous, I should say, to transport protein in other prokaryotic cell types. So these are flagella and bacteria. So in other bacteria, we see the same basic structures, proteins that are for transport. And really, if we look at all those different proteins in here, we'll notice that these structures are similar to other functioning types of proteins in other organisms or other areas. So it's not like the flagellum, all the parts had to come together all at the same time. Or I should say, it's not like all the parts had to arise from scratch at the same time. Those parts already existed in other contexts. Like here, it's not helping for movement at all. This is helping transport stuff across the membrane. But when it comes together in a bacterium with other parts and they are assembled in a particular structure, now all of a sudden it's a flagellum and it can make the bacterium be able to swim. And so exaptations, the idea there is this, where you have these structures that already exist and they are used in one function, transport, and they are then co-opted to perform another function. And so it's not like these proteins arose with the goal of having them be able to be in a flagellum. That's just a consequence of them being kind of right place, right time combination thing. So their function, though, that you can't predict the future. It's just the functionality that it has at the time, and then that's selected for, and then that may be combined into new functions later. Also, you don't know what the environment is going to be like. So the environment is also going to play a role, say current utility, because you don't know what the future will bring. So sometimes features will be selected for later for different functions because the environment is now different. And you saw that in the little video about the bird feathers being initially not for flight. We know that for sure. We'll look at a video much later in the semester about that. The structures of these reptile-like birds, dinosaur bird type organisms, are much too heavy to fly. So we know they didn't fly, but they do have feathers. So that means feathers arose and they were selected for some function because they attracted mates, because they allowed them to be warm. Who knows? I mean, we have hypotheses and they can test those with fossils, but we're not sure right now. And then later, they were co-opted and they became an exaptation because then they started being used for gliding and then flight became an advantage. And here again, not goal-oriented. So it looks like there's this kind of progression towards the complex eye of the squid, and these are all in mollusks. So you have limpets, which are sort of like a snail, but with not a curly shell, with like a domey sort of shell, and it has these photoreceptor pigmented cells, so that's what detects the light, and it can just really detect light at that point. And then the cup is forming, so it's helping, in that case, the light's now being more directed. And then you have fluid-filled cavity, so that the light is now bending in different ways, and then lenses to focus the light even more. So the image becomes an actual image by the time you get down here, versus here it's really just detecting of light versus dark. And so you might think, ooh, wow, limpets, this structure arose with the goal of getting to a squid eye. But remember, that's not true. The limpets still have their eye, and it works just fine. So there's not really a trajectory. It's just that what arose did, and so we can see that kind of stepwise progression of different features being adapted in the future for new functions. And you also have to be careful when you're looking at modern day organisms and thinking that you might be misled to think that there's a trend towards everything going towards what modern day organisms look like. So for example, the horse here, you can see that it has a hoof there and it's like one toe. So you might think that in the past, all the horses were progressing, and each next generation and the next species that arose and the speciation events had fewer and fewer toes, and then it went towards the one toe, and that's the goal, the trend. But really that's not true, right? We have multiple toes here, and then here, and then now we have one toes on these guys, but it's not like a progression of less and less and less toes as you go along. So you have to actually look at fossils in a broader context, and that's why when you have very few fossils of a lineage, you have to just be careful of how much you interpret, and that you don't go overboard with projecting what you think is happening. You have to just use the evidence that's there and make the conclusions you can make, and ask the questions that you can't make the conclusions on, and find data that help you figure out if those ideas are supported or not.